This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 383, recorded on April 1st, 2016. And just kidding, we're not actually doing a podcast. It's April Fool's Day.
Arg.
Do we celebrate this globally, or is it just the U.S.? April Fool's Day. All right, nobody can talk since I didn't introduce them. They're so good. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today here in New York City, Dickson Despommier.
Good afternoon, Vincent.
Dickson, is April Fool's Day a United States thing?
It definitely is a United States thing, but I think it's shared by other places as well.
Yeah. And
there's been some remarkable April Fool's jokes.
So I saw today that Tesla announced a cheap car. Is that an April Fool's joke?
I hope not.
No, I think it's real.
$35,000.
Is that a fraud car also, Vincent?
It's got a big battery.
It's got a gigantic battery.
Yeah, I don't know what they're going to do with that. But
you think a battery is worse than emitting
I don't think anything.
VOCs?
I think nothing, Dickson.
Oh, you don't?
I just think about viruses. So I don't know about batteries. Joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. Sorry I already blurted something out.
Nah, it's okay. It's fine.
He'll edit.
What's the weather out there like? Is it good?
It's some blue sky, some puffy clouds. I would say a little more cloudy than blue sky. And the temperature is not forthcoming. Come on.
Oh.
The temperature is 45 degrees, feels like 40. And so
chilly.
In Celsius, that's 8. Yeah.
So here we have the same sky thing with the clouds, but it's 21 Celsius.
Yeah.
It's mild today, right, Dickson?
It is, but
very nice.
Kathy's weather is heading our way.
Is there going to be a snowstorm here, Dickson?
They claim on Sunday there'll be some snow flurries.
I don't believe it. I don't believe
Kathy, how's the daffodils doing where you are?
I saw some blooming yesterday for the first time.
Bingo.
Yep.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here.
Have you had a good week, Alan?
It's been okay.
All right.
Some minor annoyances, but the weather is kind of crummy. It's warm, but it's drizzly and low overcast, and so it's 18 Celsius right now, which is 65 Fahrenheit. So that's nice, but it's supposed to be kind of rainy and damp all weekend, and then snow.
Yeah.
We have a special guest today coming to us from the lower half of the Western Hemisphere, and it's from a country we've been talking about quite a bit lately, and everyone can guess that country is Brazil. He's someone whose words we have heard here on TWiV. He happens to be an infectious disease specialist and a professor at the University of São Paulo, Brazil. Esper Kallas, welcome to TWiV.
Thank you. It's a pleasure to be here. You guys have a fan in the show today.
Yeah, I hear that you were introduced by Dave O'Connor some years ago, right?
That's right. He came to a show to talk about GBVC. In his work he's been doing in Madison, Wisconsin, and he announced to the entire group of collaborators. So I thought was important to hear that. I got addicted to TWiV since then.
Great. That's good to hear. We've had a couple of great letters from you, which we've read on the show. We decided to bring you in and hear it right from you, what's going on there, and we'll have an all Zika all the time episode of TWiV.
It's going to be a pleasure.
This episode is sponsored by the American Society for Microbiology. They want you to know about the Agar Art Contest. You should submit a photo of your agar-based art and a description of what it is or what you think it is by Friday, May 6th. Winners and select runners-up will be showcased in a gallery at the ASM Microbe meeting, which is in Boston this June. And winners will also get a free ASM book of their choosing for the complete guidelines, terms and conditions, the end-user license agreement, all that stuff that the lawyers write. Go to bit.ly/agarart2016. Esper, what's the weather like down there?
Sure, I think it's my turn to take Rick Condit's role to annoy you with the weather then. So here is 26 degrees Celsius. I'm not sure what this means in Fahrenheit because we don't care about Fahrenheit anyway. And you have scattered clouds, humidity 73, feels like 26 degrees, and it's very, very beautiful and pleasant day.
So you're not actually in São Paulo right now, right? You're outside of the city?
Yeah, I'm in my hometown. It's called Itajubá. It's in the south of state of Minas Gerais. I came to see my folks over the weekend, and it turned out that we had good connection here.
Wonderful. In general, how's the internet in Brazil? It varies?
It's pretty good.
Yeah.
It's pretty good. In big cities, you don't see many difficulties, but there are some small towns you have slow connections, but you can be connected all the time. If you can get wired internet, you can also get into the 3G or 4G networks. Should be okay.
Yeah, you sound pretty good. A little echoey, but that's fine. We would like to know where you're from. I guess you're from Brazil originally. Tell me this name, Esper Kallas. It's really unusual. Where does that come from?
So my dad was born in Lebanon and came here in 1954. So this is a Lebanese name. And,
okay.
My last name, my family name means a painter. So so that's it. So
where were you born?
Here in this very same city.
Where you are right now.
Yeah.
And you went to college and medical school in Brazil, I presume, right?
Yeah, I did medical school here, then went to Sao Paulo to do my residency in internal medicine and infectious diseases. So then I got into the graduate program of the Federal University of Sao Paulo. And when I was doing my research on my thesis was based on seroprevalence of HIV in inmates. So we went to this big prison in São Paulo City to find out in 93 that 13% of prisoners were HIV positive. It was like a very rough time back then. And for the PhD program, I was introduced to someone in the United States. His name is Tom Evans. He's an MD and also ID specialist. But also he was focused on immunology of HIV vaccine development. He was based in University of Rochester, New York, and from 96 to 98, I moved up there, stayed 2 years with my wife, and I completed my PhD experiments up there through a Brazilian program.
So do you see patients now as well?
Yeah, basically, I split 50% of my time between the lab and the patients' clinic.
What does your lab work on?
So we have a lab at the University of Sao Paulo. This is mostly a cell immunology laboratory. We have a few quite decent flow cytometry machines and ELISpots and ELISA platforms. What I try to do is bring some of the observations we do in clinical trials into the lab to do assays in human samples. So we did some contributions over the years in few areas that I think are they were decent. The first one was to demonstrate that GBVC, another flavivirus, also induces modulation of T-cell activation in HIV-infected people. And the second thing we did was we engaged as one of the sites in the pre-exposure prophylaxis landmark trial called the IPREX. This study, I think you discussed several times in TWiV, was a study conducted in 8 countries, 11 sites, coordinated from San Francisco by Bob Grant, very bright and intelligent, smart physician. So he pulled out this trial with 2,500 people to prove that HIV prophylaxis can be done using a single pill of intercytobin and tenofovir. This was published in 2010, and it's now standard practice. So several countries have already adopted, and we are at the brink of adopting here in Brazil as well. So this is something that we had the opportunity to participate in. I'm very proud of. And finally, we've been engaged in the clinical development program for the new tetravalent live attenuated vaccine that was conceived in Steve Whitehead's laboratory at the NIH. This is a vaccine that's looking very, very promising. So the NIH passed along the rights to use this vaccine to the Butantan Institute. This is a public institute for vaccine and anti-serum development in the state of Sao Paulo. They got Steve's vaccine. He made a very important step into the vaccine as transforming it from the liquid form to lyophilized form, which is very, very more convenient for clinical studies and eventually, if gets approved, to be used in the field. And then after that step, we were able to put together a phase 2 clinical trial. So from the University of Sao Paulo, I did coordinate this trial since 2013. It's ongoing. We're wrapping up the data. So news to come in the next month. So we're analyzing the data, probably to have it out around May as a communication. So very exciting times. But because the other data from this vaccine also turned out to be very, very positive, including one paper that just came out at the Science Translational Medicine by Ann Durbin from the Johns Hopkins in collaboration with Steve Whitehead, proving in the human challenge model with dengue-attenuated dengue 2 strain, they were able to demonstrate 100% protection. We convinced local agencies to step forward with the phase 3 clinical trial that started in February 22nd. This is a 17,000 clinical trial in 14 sites around Brazil, and it's going to randomize 2-to-1 vaccinees and placebos, and we're also going to enroll people from adults 18 to 59 years old, older children and adolescents from 7 to 17, and children from 2 to 6 to be followed, trying to demonstrate whether this vaccine is efficacious in protecting against all 4 serotypes of dengue.
This is TVO03, right?
Exactly. It changed the name just because of the way the vaccine is now lyophilized, but it's basically the same product.
Okay, so and this is of dengue vaccines that people have tried. This is the one furthest along in clinical trials right now, isn't it?
That's correct.
Right.
After CID from Sanofi,
right.
That has been the studies have been completed.
So I think it's been licensed in a few countries also, right? The Sanofi vaccine.
Yeah, it was licensed in Mexico, Brazil, and I believe it's Philippines, third country.
That's called Dengvaxia, right?
I think it is, correct.
Right.
Right. We talked about that. I think they were mostly all licensed around December 2015.
Yeah, right. Yeah, we have an email from someone today who sent us an article about this new vaccine, and the article is actually written by Karina Storrs.
Oh, yeah.
And it's on CNN, and it's all about TWiV 005, and they mentioned Steve Whitehead, and it's how good it is. I was going to actually get the original article and do it in a couple of weeks, so that's pretty interesting.
You know, TWiV has one minor difference, because when Steve did the preliminary studies, he realized that the second component of the vaccine, the dengue 2 vaccine, was not the entire dengue 2 virus. So he made a chimera from dengue 4 attenuated virus and inserted the dengue 2 envelope. So he always been quite suspicious whether the dengue 2 component could be a little bit less efficient in performing in delivering the protection.
Yeah.
So they came up with the TWiV, that the only difference is that the dengue 2 component, in this case this dengue 2 chimera, instead of having 10 to the third particles per dose, has 10 to the fourth.
Mm-hmm.
And it gives a little more boost in the dengue 2. But I mean, we've been working with the TWiV. All 4 components have the same dose, and the results will tell us whether in the phase 2 trial the immunogenicity could be comparable.
Yeah, because I remember that was a problem with the previous dengue vaccine. The 2 component wasn't performing as well, right?
Correct.
So,
And
that's the predominant serotype globally, isn't that correct?
Well, it depends on the season, because this year Brazil had the worst epidemic of dengue, 2015, and we had 98% were dengue 1. So it keeps changing from year to year. So,
were there weather patterns that favored that epidemic?
That's a good point, Dickson, because we really don't know whether weather would change dengue spread in the country. And I believe there is a role, because last year we had a drought.
Yeah, that's what.
And was very unusual. So we had lots of dengue. Dengue never had visited the city of São Paulo as it did last year. Surprisingly, this year in the city of São Paulo is much milder, but there are other states that it's coming up. So I think it depends on the local weather. You're probably right about this, but there is no explanation why the dengue would be higher or lower in certain areas depending on the kind of climate, Dickson.
Right, and also malaria, I guess that must have been affected too, but maybe in a negative way, do you think?
Well, malaria is limited to the Amazon basin and some small scattered points on the coast of São Paulo state. So we don't see that same effect. And when malaria happens, it's humid all year long.
Exactly.
Most of the time,
So Esper, can you give us a Zika report?
Sure. So let me start by the last numbers. How about that?
Sure.
So the Ministry of Health have been collecting numbers from Zika cases all around the country, but most of the report has been concentrated in the microcephaly. And this is something that we can discuss later, because microcephaly, to many who are now seeing cases or have access to published data, seems to be just a proportion of cases as a consequence of a Zika infection in a pregnant woman. But in regard of microcephaly, the grand total cases we have reported in the country is 6,776. And every case that comes in, it poses a major challenge to the system, because you need to rule out all other causes of neural defects. And it's a long array of causes, including the torch syndrome and congenital diseases and alcohol abuse and several other causes. So in most of these cases, they are still under investigation. So we have 4,291 that are under investigation. And among those, 2,485, the investigation has been completed.
Now, these are, just so we're clear, there have been a lot of numbers coming out related to microcephaly, and there's an issue with diagnosis of microcephaly, where when those are subsequently investigated, in many cases they turn out not to be ruled as microcephaly when analyzed by experts.
Exactly.
So are you now talking about the cases that have been definitively determined to be microcephaly, and now we're asking whether there was Zika virus present, or are we still on the microcephaly question?
No, we're still in the microcephaly question. So
okay.
Let me go back to what you just mentioned. So Brazil didn't have a good reporting system of microcephaly, and the numbers tell you that. So if you look in 2014, the country reported 157 cases of microcephaly. United States had around 20,000. So we're way below. I mean, this is a demonstration of underreporting, massive underreporting. And the other problem is the reporting system Brazil have adopted was an old system where the cutoff for microcephaly was very sensitive at 33 centimeters of head circumference. And that is exactly the 10th percentile, meaning that one in every 10 children would fit the criteria of microcephaly based on that criteria in the threshold we adopted back then. So when this microcephaly story started, there was not a single woman in the country who did not measure his son or daughter's circumference. And what happened immediately after it hit to the press, there was a flood of microcephaly cases.
Right.
In many of those cases, these are normal children falling into the 10th percentile. And then after they realized there was a mistake going on, they revised twice this classification. Now the classification is meeting the same criteria WHO has been adopting around the world. So then we have, in the beginning of this epidemic, of a over-reporting, for sure, and now it's been adjusted. So the numbers reflect a little bit of that. So if you look at the numbers with the investigation that were completed, it's about 2,500 cases. Only 38% of those cases are possibly associated to Zika. And when I say possibly associated, means there is a neural defect that's confirmed, other causes have been ruled out, and the mother reported a potential acute febrile illness during pregnancy.
Okay. Now, earlier statistics that I saw had suggested that over half of the cases that were suspected microcephaly that were investigated were discarded as not being microcephaly at all.
Yeah, so it's 62% now.
Okay, so 62% of the cases were discarded as not being microcephaly at all. And then 100% of the remainder are possibly associated with Zika virus?
That's correct, yeah.
So there aren't any cases that have been confirmed as microcephaly where you know that it was where there's CMV as a possible cause, or where there's alcohol as a possible cause, or any of the torch pathogens.
No, I think these cases, they are pulled into the discarded cases. So there are cases now reported, you see?
So the discarded cases are not discarded as being microcephaly. They're discarded as being Zika associated.
Yeah, or not Zika associated, or they have another cause, or it's not a microcephaly case.
It's both. Yeah.
Okay, so that's all lumped together into the discarded cases.
Yeah, so torch, the T stands for toxoplasma, and that would actually result in a macrocephaly or hydrocephaly.
Right.
In most cases. So that's pretty easy to distinguish. But the rest of them, I was not aware of microcephaly as a sequelae to some of those viral infections.
It could happen, I think.
Yeah,
especially rubella.
Yeah. Okay.
Now, the other thing is, early on in this, when this became big news and we started talking about it on TWiV, it was the microcephaly link has been obviously the big cause of all this coverage, I think. And one of the things that jumped out immediately was that historically there had been this tremendous underreporting, and that was obvious when you looked at the numbers. There couldn't possibly be that few cases of microcephaly. And then what was reported as a so-called epidemic of microcephaly was a 20-fold increase. Mm-hmm. But if you increase the historical numbers by 20-fold, you actually just got into the ballpark of the rates that are considered normal in the United States and Europe and places with more robust reporting.
Sure.
So have we nailed any of that down? I mean, do we have any indication that there really is an epidemic of microcephaly, or have we just eliminated the underreporting?
No, I think we need more data to be consolidated, but there is no question anymore that there is an increase of microcephaly cases that are related to the Zika infection. And let's look back to the numbers. So out of these 944 cases, they are still under investigation for Zika, or they're still processing samples, but they were able to identify the Zika virus by PCR in 130 of those.
Right, but if you randomly sample the population in Brazil, you'll identify Zika virus in a number of people.
You're right. I mean, this is something that people have raised as a potential confounder. So many of these microcephaly cases were from women who acquired the Zika virus during pregnancy and was there just visiting the fetus for any other reason. But the evidence of association is based on the case reports that we now have published. And so the conclusion that it could be a microcephaly epidemic is based on these increased number of cases related to acute febrile illnesses. As you said, the PCR identification in the samples, but also the potential correlation established in these case reports that have been coming out.
Okay.
Esper, so you said 130 cases positive for Zika. Is that the mother or the fetus?
That's the fetus.
Okay.
Now, one of the things that we've talked about is, I'm sure you've heard on TWiV, is the possibility that Zika virus is just opportunistically infecting the fetus in a pregnancy that's already gone bad.
Yeah.
And I suspect that there's not going to be a way to distinguish that for a very long time.
I think the only way, Alan, is if we have very rigorous case control cohorts.
Right.
So we need this. These are undergoing. There are several sites that are pursuing this. There is a site in Natal, northeast Brazil. There is a group in Ribeirão Preto where Vincent has visited before. They have a cohort put together there. There is a cohort in Rio underway that we know. And we have a study designed for São Paulo, but the Zika epidemic did not reach the city yet.
Right.
But these studies are ongoing.
Good.
Some other numbers here maybe you want to go through.
Sure. So we had 208 deaths, and out of these deaths, 47 of them had Zika isolated. So in this case, they represent 68% of the deaths related to microcephaly that were investigated. Here, the Zika virus isolation represents, a big chunk of those cases, suggesting that there is this correlation is real. And I also brought to you a picture of a case.
Right.
It's if you look at this kid. So this picture was taken by a colleague. His name is Drauzio Varela, a very known MD that does also a TV show here every Sunday. And this case shows us a few details that are very interesting. This
now,
kid was,
I'm assuming this is not one of those cases where they would say, "Gee, is this microcephaly?" This is a pretty definitive case, right?
Oh yeah.
Okay.
So this kid unfortunately was born with the severe defect. I can show you the CT scan later, but it's very compromised encephalus with a lot of calcium and enlarged the ventriculi. And what you see here is something very interesting. So you see that the skull is shrunk. And what seems to be happening in many of these cases, what we see here, this head at some point was a little bit more developed. But at some stage of the pregnancy, there is the neural system is attacked by some factor that we believe is the virus and destroys the neurons and glial cells. And what happens is the encephalus volume decreases and the skull bones, they collapse onto the head. So what you see, this convoluted skin of the head, it represents one of these aspects, that this kid suffered more late into the pregnancy a very fierce attack by the what we believe it's the virus.
So is the skull also convoluted then in this case?
No, I think the bones they fall onto each other. because there is nothing. I mean, the basis of them, which is the encephalus, it shrinks. You see.
Right.
I see.
And this is as a newborn, the bones will not have fused. They'll just be, the plates will just be kind of sitting there on top of what's left of the encephalus.
Exactly.
Wow.
So the other characteristic of these babies, most of them retain basic reflexes, meaning that the stem of the brain is preserved. So they can suck and sometimes feed themselves. The more severe cases, they do not, obviously. But in many of these cases, despite the severe damage to the upper brain, they retain the functions of the stem. We don't know how long this is gonna take. I mean, if it's gonna deteriorate over time, but it also is in line with the observations from these case reports that are coming out, 2 of them in the New England Journal of Medicine, that the virus seems to preserve the stem of the brain.
Now, is that typical of severe microcephaly, or is that unique to cases that are associated with Zika virus?
So, Alan, I must confess that I'm not a specialist in brain development and fetal development. So I have no long track record in following other torch causes of microcephaly. But the people living in this area who work with children development says that this is a quite unique characteristic of this Zika-related neural defects.
Do we know in any of these children who have been born like this individual, has anyone looked for virus? Do we know if it's present or not?
So the virus has been searched for in all of these cases, and by the numbers,
that's the number. 130.
130 were positive.
Right. So now, I guess there are 3 possibilities here. Either the virus is directly and independently causative of this microcephaly, or it's worsening a microcephaly that had started already, and that's what allowed the virus to get through the placenta, or it's just there as a bystander, and perhaps some of the pathology here, if it really is distinctive to the Zika associated cases, could at least get us to narrow that down to 2 possibilities.
Sure. I think these are the 3 working hypotheses since the beginning.
Right.
So, Esper, you had said hasn't reached Sao Paulo yet. Is it expected that it will?
We believe it will, because we have dengue. So why not having the Zika?
Right.
Why it did not come to the big city, we don't know. Perhaps it's related to the fact that the dengue season 2016 has been quite mild, and there got to be, as Dickson mentioned, an unknown climate factor that's preventing the virus to come into the city with the same force as that has been seen in other big cities in Northeast, especially. What is this factor? We don't know. It's going to enter the city of Sao Paulo this year. We don't know. There are people suggesting that this is just a matter of time for this season. Some suggesting that it's going to be in 2017. But I think it's just gut feeling, and I think we need to wait for data.
Now, you're presumably Sao Paulo monitors the mosquito populations, right?
Does, yes.
So you've got the vector there.
We do. Aedes aegypti.
Right. And I guess the question is just getting a critical mass of people infected.
Yeah, I think that's what we're expecting to happen at some point.
Right.
But until we are now early April, Fool's Day here as well.
Yes.
And there is no confirmed autochthonous cases in the city of Sao Paulo.
Mm-hmm.
And there are no reservoir hosts.
Well,
maybe
we have lots of Aedes, and most of the people in the city are susceptible. And surprisingly, we have a very large migration of people coming in and out of the city from all the areas of the country. São Paulo is the biggest hub in the country. One would expect that having people coming here happens. Yeah. In fact, there are a few cases of women who were pregnant and had babies with neural defects related to Zika in the city, even isolated the virus. I mentioned to you one case of a lady who had Guillain-Barré syndrome we cared for in the city. So the people with the virus are coming in, but for reasons we don't understand, was not necessary to trigger the transmission in the city.
And in terms of reservoir hosts, there are New World primates in Brazil, but I gather nobody has yet found any evidence that Zika can infect New World primates.
Yeah, I'm unaware of this this type of
So it may end up being, I guess, dengue is kind of like this in Latin America as well, where it's strictly an urban cycle. It doesn't have the sylvatic cycle that you see in Africa.
Yeah.
I believe I've heard of it infecting howler monkeys. And I guess I thought howler monkeys were in the Western Hemisphere.
They are, of course they are.
Okay. But I can't place where.
Howler monkeys.
Yeah.
Oh, yeah. They're all throughout Central and South America.
Right. So this is, there's been a die-off among howler monkeys.
Yeah, that's right. We brought this up, right? Remember
yes.
that?
Okay.
Yes.
In Nicaragua, right.
It's not clear whether it's Zika or whether it's something else, but
Right, okay.
Oh, yeah, a ton of people looking at that.
right.
University of Michigan, in fact, is looking at it.
Is that Aubrey Gordon?
Cortez, Ortiz, and Williams Guillen.
Okay.
Yeah, so they're looking at this to see if it's Zika or something else, which they should know by now, I would guess, because this is all from back in February. Yep.
Right.
I remember doing that. Yep.
I'm sure somebody's got an answer.
Okay, Esper, anything else? I'd like to talk about some papers, but if you have some more things you want to bring us up on.
We can bring up during the paper discussions.
So there have been, as every week, there are a ton of papers coming out. And one that I particularly liked was a paper published in Science called Zika Virus in the Americas, Early Epidemiological and Genetic Findings. And this is, what is there, about 50 authors on this paper.
Yes.
And if
Looks
I
like
tried,
a physics paper.
if I tried to pronounce them, I'd get them all wrong. And I don't want to do that.
I can't help you on that.
Or maybe just do the first and the last, since there's so many.
Nuno Rodrigues Faria.
So Nuno Rodrigues Faria is a Portuguese scientist. He is based in Oxford, but he also has a second appointment at the Evandro Chagas Institute at the north of Brazil, state of Pará, one of the hotspots for Zika. He works in the laboratory of Oliver Pibas at the zoology department in Oxford. Very bright scientist. The last one is Pedro Vasconcelos, who is the senior investigator at the Evandro Chagas Institute in Pará.
Wonderful.
Cool.
So this is a paper. They collected 7 Zika genomes from Brazil and did some, I shouldn't, the word that Eugene Koonin doesn't like, let me stay away, computational biological analysis of these to figure out, how they're related and the common ancestor. So they do phylogeny, they do molecular clock analyses, and the results suggest that these have come from a single introduction of the virus into the Americas sometime between May and December 2013, which would be over a year before the virus was detected in Brazil,
right,
which I think is very cool. They don't find any changes in these genomes that they can correlate with, microcephaly. They say in several places, which I really appreciated, there's no reason to posit that this lineage is associated with increased disease severity. They say that a few times in the paper, and a lot of people have been publishing and talking about how, these strains are mutating, and that's why they're doing this. There's no evidence for that. Here we have people who know what they're doing, and they're saying there's no reason to say that any of these changes mean anything.
And these are most closely related to the Pacific Island.
That's right.
Strains, suggesting that they came over from. So there was the outbreak on Yap, and then that got through Polynesia, and then
Right.
that is probably the point at which this was introduced into Brazil.
So they do a molecular evolution calculation, and they say this rate, although this rate is high compared to whole genome rates for other flavies, it's consistent with retrospective analyses of previous estimates. Hence, this result should not be interpreted as implying that Zika in the Americas is unusually mutable. And what happened in the Ebola outbreak last year, people were saying Ebola is mutating. And
of course it is. It's a virus.
It is. So they estimate the date of the most recent common ancestor of all the Brazilian genomes is August 2013 to April 2014, and the common ancestor of the French Polynesian and the America lineages is December 2012 to September 2013. Now, here's the cool part. They want to say, can we figure out how this virus got into Brazil? So they collated airline flight data from all countries with reported Zika outbreaks between 2012 and the end of 2014, which is roughly when this virus might have come in. And from late 2012, they find an increase in the number of travelers arriving in Brazil from 3,775 a month in early 2013 to 5,754 passengers a month a year later. And this increase coincides with the period during which they think the virus entered the Americas. So if the epidemic did arise from a single introduction, which is what they're saying, then it must have been circulating for at least 12 months before the first case was reported in May 2015. Now they go to these hypotheses. There are two published hypotheses for how Zika came to Brazil. One during the 2014 World Cup, which is June 12th through July 13th, or how do you say this, VAA canoe event? Va'a?
Yes, that's Polynesian.
Because Esper is not going to help us with that one.
Pretty sure, yeah.
But it's in Rio anyway. The Va'a canoe event in Rio between 12th and 17th August 2014, or they say could have occurred during the 2013 Confederations Cup soccer tournament, which was in June of 2013. So they say the second and third events had competitors from French Polynesia, so they're appealing because the virus was there. But they say our results indicate that the introduction predated the first two events, the World Cup and the Va'a event. And so it's not clear when it came in. They say, this is very nice, we believe that large-scale patterns in human mobility will provide more useful and testable hypotheses about viral introduction and emergence than ad hoc hypotheses focused on specific events. But don't blame it on the World Cup.
You can blame it on the Confederation Cup.
Yeah. You can blame it on the Boeing Corporation.
Well, the fact is that probably many people brought it in, but only one took off, right?
Yep.
Yeah.
That's it. I'm sure.
But also, the increase in passenger traffic could correlate with simply increased trade between the two countries, which would also be done by ship.
Right.
Yeah.
One load of tires can easily introduce this, or a crew member on a ship.
Sure.
Do we know whether human viremias can get high enough to allow transmission by being bitten by a naive mosquito? Because those are the same issues were raised during the West Nile virus outbreak in this country.
Wasn't that how it spread?
Yeah, but they claim that titers for this virus rarely reach high enough limits in adults. So this must have been a child that introduced West Nile. So I just wonder if Zika had similar pattern.
Well, but in the case of West Nile, you have reservoir hosts flying around all over the place.
Yeah, but it wouldn't have. Well, that's true. That's after it gets here, though, Alan. I just meant that. How did it actually? What was the first introduction?
Okay.
Yeah. So you're saying if the viremia isn't high enough, a mosquito would not be likely to pick it up.
Yeah, that's right.
Unless we're a kid, right?
Yeah, exactly.
So that could be the same for
and children. Many children do not get sick from high viremias with West Nile. So I just wonder what the clinical pattern for Zika, which is a milder flavivirus infection.
Right, but in the case of Zika, we're routinely seeing human-to-human transmission. I mean, that seems to be its normal mode here. And so obviously it is possible for a mosquito to pick it up from the bloodstreams of infected people.
Sure. Then you raise the question, why is it so non-pathogenic then if it reaches that level of viremia without causing much disease?
Right.
Well, we do have some numbers on Esper. Do you know offhand the genome copies in the blood of?
We usually see here in the cases we have the opportunity to run RT-PCR that the viremia is not very high. It stays around the thousands or the 10,000s, but there are a few cases at 100,000s, especially the closest you are to the onset of symptoms.
Mm-hmm. So maybe that's why Tahiti didn't do so well in the Confederations Cup. So when Oliver was interviewed by the Nature reporter, he believes that potentially the game that was the culprit for this introduction of Zika was a match that occurred in Pernambuco, which is one of the biggest hotspots for the Zika epidemic and the microcephaly epidemic, when Uruguay beat Tahiti 8-0. And actually, Tahiti had a very, very unfortunate set of results during the conference. It played Nigeria, lost 6-1, and then lost to Spain 10-0, and then to Uruguay. But the Nigeria game was in Belo Horizonte, which is southeast Brazil. Yeah. The Spain game was in Rio de Janeiro, which has now been hit by the Zika epidemic. And the third game, the Uruguay game, was in Recife. So he believes that was the gathering that brought someone who was infected from Polynesia to be fed by a mosquito in northeast Brazil.
I don't know. I think in this paper we read that would be an ad hoc blaming of.
Yes.
That's what they're warning us not to do. But it's interesting anyway. But we don't know if that's the case.
Yeah, it could also have been somebody who was, random business person or what have you.
Yeah. So some other papers that came out. Some of these I will pass through quickly, and if anyone wants to pause longer, please feel free to do so. In a paper in Medicine, investigation into an outbreak of dengue-like illness in Pernambuco, Brazil, revealed a co-circulation of Zika chick and dengue virus type 1. And this is a paper coming out of São Paulo, right, Esper?
Yeah, it's from a collaboration between the University of São Paulo and the colleagues in Northeast. Sabri Sanambani is the last author. He's a virologist who works on virus sequence.
So here go ahead, sorry.
This is not terribly surprising. I mean, we knew these viruses were all circulating there.
Yeah. Yeah, so this is basically showing that all three were there at the same time. And then they had some co-infections, I think, of two viruses at a time. And
there's been this weird pattern that I think we've commented on before, that Zika is basically following chikungunya around the world.
Right.
The chikungunya went to the Indian Ocean and across the Pacific, and then got to Latin America and the Caribbean, and now Zika virus is following the same type of path.
Yep. Of course, you go around the world, it's the only way to go, right?
It's right. You're either going to cross the Pacific or the Atlantic. So what are your options?
Not sure what that means.
Have we had any cases in any of the teams that played and then went back to their own countries?
Good question. I don't know. Do you know, Esper?
No, not really. No.
I don't know. Okay. All right. Another paper which I think is pretty interesting from the American Journal of Tropical Medicine and Hygiene.
My goodness.
Which, I'm happy to see this here because we don't do too many papers in this journal. Mm-hmm. It is characterization of a novel murine model to study Zika virus. The first author is Shannon Rossi, and the senior author is Scott Weaver from University of Texas in Galveston, also New Mexico State University in New Mexico. And here, what they have got is immune-compromised mice lacking either type 1 or type 1 and 2 interferon responses. You infect these with Zika virus, I believe subcutaneously. They develop a viremia. You get virus in the blood. You get virus in the brain. You get neurologic disease. You also get robust replication in the testes of these animals. And if you infect young mice, they die. If you infect older mice, they still get illness and viremia, but then they recover.
It's a pretty good model.
Yeah, the only problem is you're taking away the innate immune response.
Sure.
So if you want to look at true pathogenesis, that's going to be a little hard. As they say, in fact, in this paper, the use of immunocompromised mice is not ideal for describing the pathogenesis of Zika. But maybe if you want to test antivirals or vaccines, it might be okay to do that. Now, what I found interesting here, they do an experiment. The first experiment they do, they just infect wild-type CD1 mice subcutaneously with 10,000 PFU, no illness, no viremia, apparently no replication. And so that's why they went to the immunocompetent mice.
Now, back in 1947, when this thing was first discovered, the initial isolation was done by taking the virus from the monkey that was the sentinel monkey that came down with it, and passaging it in mice, right? But that was done with something like intracerebral inoculation.
Yeah, that's it. I think that's the key. They do IC, and all the mice die.
Right.
But it probably is not doing much elsewhere, I would guess, unless a little bit is. So it's not a great model.
Not a very useful model, no.
But subsequently, someone did passage the virus, I think, 17 times, and you get a mouse-adapted strain, and then you can put that into wild-type animals, and they get disease. But, that's got its own issues.
It's a different virus, yeah.
I went and looked at that paper, and they have these lovely charts where they infect 7 mice, and each mouse gets a little circle. And then they show what happened to each mouse by a dot or something, and then they show which mouse they took virus from to pass on to the next set, and they had 17 of these lines. And it's a kind of image nobody makes anymore, but it's so informative because you could look and see which mice were okay, which got sick, which died. Yep. And you can see by the filled-in circles, which is death, as you pass the virus, it gets more and more lethal. The visual representation of results.
Yes.
A lost science. Anyway, this is a cool paper. I think that's nice. Okay. Now, this is something that I would like to get Esper's thoughts on. We have a paper. We've talked about this before, so this is just another one. Getting infectious Zika virus from saliva. And I have so many papers here open.
I just went to the mosquito, to the Aedes albopictus
Is that
one.
before that? Maybe, yeah, sorry. We don't have to do them in any, but I'll do that next. How's that? Okay, here's a Eurosurveillance paper. Isolation of infectious Zika virus from saliva and prolonged viral shedding in a traveler returning from the Dominican Republic to Italy. This is a 20-year-old woman, and she shed virus in saliva and urine for up to 29 days after symptom onset. So we've talked about this fact before, that you can get virus from saliva and urine. That has good diagnostic utility. But the question that's interesting to me, and which is actually addressed in a kind of opinion paper from Brazilian Oral Research, that's the name of the journal, and it is Zika virus infection spread through saliva, truth or myth? And they say basically, we have infectious virus in saliva, and other viruses are known to be in saliva, like cytomegalovirus and Hep B, and they're transmitted that way. But saliva also has antiviral properties, so you can't assume that just because a virus is there that it's going to be transmitted. So the reason I'm interested in this is because we have these supposed cases of sexual transmission where it could easily be transmitted by saliva, and that's why I don't think they've proven sexual transmission, although, if you ask Bill Clinton, he would say kissing is not sex, but that's obviously part of it, right? So I don't know what the CDC considers. So what do you think about that, Esper? Any thoughts?
Well, I think there is a possibility of person-to-person transmission. Sexual transmission is quite plausible based on the several case reports that are gathered, especially in the U.S. and Europe. But it's very hard to tease out between oral contact and sexual intercourse.
Right.
So we still need a good model to try to understand that. If you look at the several other viruses transmitted by saliva, you find several examples of virus containing in the saliva that are not transmitted, such as HIV, and they are found quite substantially in saliva, but for several reasons, it doesn't complete an efficient transmission.
Right.
Nevertheless, my suspicion, and again, we need data to prove this, the person-to-person transmission may add to the epidemic, but may not be as significant as the mosquito transmission.
Right.
So you might say it's a byway rather than a highway.
Sure.
Okay. All right. Let's do this mosquito paper. It's a PLOS neglected tropical diseases. Differential susceptibilities of Aedes aegypti and Aedes albopictus from the Americas to Zika virus. So this is a cool paper where they want to look at the vector competence. They basically feed mosquitoes Zika and see if it spreads within the mosquito and if it gets to the saliva. So that would be, replication and transmission potential. All right?
Right.
And they use local mosquitoes. So what do? They use Caribbean mosquitoes. And do they use South American mosquitoes as well?
Yes, they do.
They do.
Rio.
From Rio.
And the authors on this, I'm going to let Esper pronounce the first author at least.
Thais Carneiro.
Okay. And Annabella Fayou?
Yeah, this is a French name. Yeah.
Right.
So this is from Rio and the Institut Pasteur, French Guiana, Guadeloupe, and New Caledonia.
Pasteur of New Caledonia, yeah.
And the important,
I was thinking some of the mosquitoes came from New Caledonia, but
virus,
maybe not.
I think the virus came from New Caledonia. Yeah, the virus was isolated in 2014 from New Caledonia.
and the importance of this is that Aedes aegypti and Aedes albopictus have slightly different niches. So if the virus can move from aegypti to albopictus, that increases its range, as we've actually seen with chikungunya.
So they find that both mosquitoes can take up the virus, replicate it, and potentially transmit it by being in the saliva. They find that aegypti from Guadeloupe and French Guiana had higher dissemination than other Aedes aegypti populations. But overall, it was lower than they would have expected. The vector competence, they were surprised that it's so low, and it makes them conclude that other factors like a large naive population or high densities of mosquitoes and people must be also contributing to the spread, because the mosquitoes aren't that great as far as mosquitoes go for replicating and transmitting it. Which,
if it's borne out, would be great news, because there may be some critical threshold.
Yeah.
If you really need high densities of infected people, viremic people, and mosquitoes, then maybe even the initial efforts that we make at mosquito control will push us past a threshold and the disease will burn out.
So, Dickson, does it surprise you that mosquitoes in French Guiana and Guadeloupe are different from South American mosquitoes? That the same species?
No, not at all.
And how do they differ?
Well, they differ in probably their temperature tolerance for ideal reproduction rates and biting preferences and maximum numbers of eggs yielded per blood meal, etc. I mean, a whole bunch of biological parameters you find different, because these are all island communities for the most part, and you get rapid degeneration of the parent species and adaptation to the island species or island life cycle. I just saw a program last night, which is not irrelevant for this discussion, of release of pigs on islands. And the smaller the island, the smaller the pig. Because eventually they'll adapt to the size of the environment that they're in by simply reducing their caloric intake, and because it's less available and therefore you survive on less. Selects for the ones that are smaller that can survive better, and the next thing you know, you've got a completely different pig. So that's how I think that's how life works in general.
All right. I like this paper. It's very interesting.
Yeah.
And it just shows that it's not ever simple. It's not just about the mosquito and the virus growing in it.
Exactly.
There were a bunch of good review articles published in the last couple of weeks. There is there are, 4 different ones. One by Scott Weaver, one by Helen Lazier and Mike Diamond, and another by Helen and another author. And then a 4th one, which I think Esper just pointed out to us this morning. It's absolutely fabulous. It's just called Zika virus. It's by Didier Musso and Dwayne Gubler, who are from Tahiti in French Polynesia and the Duke Singapore Medical School.
That's a fascinating review.
It's really good. It is totally comprehensive. It's like really detailed, everything you wanted to know, and everything about this virus from the beginning to the current. It's just beautifully done.
Dwayne Gubler used to run the CDC vector control program at Fort Collins.
That's right. Now, what I find cool
and
here...
I think all of these reviews are actually open access, which is a little unusual.
Yeah. It's good.
It's very good.
It's very good.
Yes.
Now, what I find is cool, so that first monkey in the Zika forest,
Yes.
that got a fever, and then they isolated Zika virus from that. It was monkey 766. And, of course, the MR766 strain is the prototype from African strain. We have it, and I look at it, and I think of that monkey.
Uh-huh.
I just think that's connected is really cool to be able to do that, right? You can't always do that. I think that's really fun.
Yeah.
Why did the virus fall out of the tree, right?
Peer pressure.
Okay, what else here? Oh, so here's a good one. Another nice New England Journal case report, which Alan and actually Kathy pointed out was coming out yesterday, and maybe also Esper. Everybody knew this. a couple of embargoed papers this week, which we heard about and we got yesterday. This one is Zika virus infection with prolonged maternal viremia and fetal brain abnormalities. Have you read this, Esper?
Yeah.
Do you want to summarize it for us?
Sure. This is a lady from Finland. She was with a family in Washington. She decided to take vacation in the Caribbean. She went to 3 places in the Caribbean, and they believe that the location she was infected was Guatemala. She developed a acute febrile illness in week 11 of pregnancy, and that went away because also she was not diagnosed at the site. But then I think she went back to the U.S. and to Finland. They did several serologies and found out she was circumverting for Zika virus by a specific IgM. She carried on the pregnancy, and down the line around week 12, despite previous normal ultrasounds, she was diagnosed with carrying a baby, unfortunately, with some neural defects. And one of the things that are interesting in this case report is the finding in the ultrasound just prior to week 20 was that the baby had a potential bleeding in the brain, and in some changes in the corpus callosum of the encephalus. And then they carried out an MRI. The MRI, and there is a picture in the paper, is remarkable because the level of detail, and they show absolutely everything was going on in that brain. And this is a discussion we had recently here in our group, because ultrasound is a very poor method to detect early changes in the brain of fetuses, especially prior to week 20. And this case is a very good example when potentially the use of MRI can sort out whether these neural changes are taking place early in pregnancy, much earlier than the ultrasound can. And the other thing, so she carried on a little bit more, decided to terminate the pregnancy based on local laws and personal decision. And they observed several brain defects. Again, these brain defects were more concentrated in the upper a part of the brain, the encephalus, and but they found the Zika virus in fetal brain, placenta, membranes, umbilical cord. But the most interesting thing, the virus was also isolated in the mother serum by the time she was terminating her pregnancy.
Yes.
So she retained the viremia throughout several weeks. Potentially, she retained viremic during several weeks, or she had escaped the virus to her blood in some occasions during her pregnancy.
Yeah, and the idea that it's reverse flow of virus from the feet, because she had cleared her infection early on, right?
Sure.
And then so that's very interesting. I also found interesting that they get virus. So the last one of the previous case reports we did, the virus was only found in fetal brain. I think that was the first time they got the entire genome from a fetus. But here we get in brain as well as muscle, liver, lung, spleen. It's elsewhere, so it's not just neurotropic, apparently.
And they actually do real virus isolation.
They get virus isolation on neuroblastoma
This is not all
cells.
PCR. They actually they get virus out and they sequence the virus they get out of neuroblastoma cells.
Yeah, so now people are using this neuroblastoma line, SKNSH, in addition to Vero and mosquito cells. Apparently it grows really well in neuroblastoma cells as well. And they also have pictures of virus particles here as well. So the husband also was Zika positive. So they both went on vacation together.
And by the way, the author, this is a really excellent paper. It is open access. It's in the New England Journal. The first author is Rita Driggers, and the senior author is Ali Vapalati.
Of course, the genotype is the Asian genotype and resembles all the other viruses from this outbreak. Yeah, so that's an interesting paper. It's very well done. Nice images as well.
Yes.
And again, they have amino acid differences, and they don't know what they mean, of course.
Right.
Yeah, that's a good one. Thank you, Esper. I think the last one we'll bring up is the 3.8 angstrom resolution cryo-EM structure of Zika virus. So we don't have to have a artist's rendition anymore. Can actually, Kathy, you want to tell us about that?
Sure. This comes from Purdue, my alma mater.
Yay.
Yeah, a collaboration between Michael Rossman and Richard Kuhn. They also included Ted Pearson, who's at the NIH. And the first author is Devika Saroyi. She's a graduate student. Actually, there's, I'm trying to see, co-first authors. The second listed author is Zhenguo Chen. So co-first author with Devika. I remember in one of the places seeing a picture of her, and then something we'll get to when I do my pick of the week. The third author is Lei Sun. The last author I haven't mentioned is right in the middle of Thomas Close. So they used cryo-EM to figure out the structure of Zika virus, and I'm trying to remember now which strain of Zika they used. They say right off the bat somewhere.
Polynesian strain,
yeah, that's right. Yeah. And so it looks very much like dengue virus. And this group has a lot of experience with doing structures of dengue virus, so they should know. And the interesting thing is that there's a change in the, it's in the M protein or the E protein?
E protein, right.
The E protein, right, that has this, when you superimpose structures of other flaviviruses, there's this loop that's different for the Zika virus, right. And that has to do with a difference in glycan binding. And so they have structures that actually even show the difference in the glycan. So it's really nice tour de force. Richard was just here last week, and I should have asked him, when did they start it?
Did he talk about it when he was there?
No, he wasn't giving a seminar or anything. He was just here for a visit. But yeah, so this.
Well, if you're set up for cryo-EM and you're good at it, I gather it's not that big a deal.
Right.
you need to get the protein or the virus, but then once you have it, it's just a question of crunching through and putting things together. Not to minimize the amount of work that went into this, but this is obviously a team that has.
Oh yeah,
this was not their first rodeo.
And I think based on the Purdue press release, there was some new equipment involved that helped facilitate this.
One of the figures looks so beautiful, you could make a stained glass window out of it.
Go for it, Dickson.
Yeah. Mm-hmm. The one that's colored blue inside with the
so the cool thing about these flavies is they have a capsid on the interior, proteinaceous, where the genome is encased, and then a membrane, and then the glycoproteins in the membrane, and the glycoproteins assume some of the underlying icosahedral. They assume the underlying icosahedral symmetry as a consequence of having that capsid. It's beautiful. It's just gorgeous. Yep. On Wednesday, so this paper came out Thursday. On Wednesday, I asked someone in my lab, Amy, remember, Dickson, you were here. I said, when do you think the cryo-EM of Flavie will be out? And I was just, fooling around. She said, tomorrow.
Yeah, she did. She just flippantly said tomorrow, and the next thing you know,
I wrote to Richard Kuhn yesterday, and I told him that, and he said, did you have some insider information? No. I figure you guys are going to crank this out first, right? Because you're always doing it. It's just beautiful.
It is.
It's absolutely beautiful. So a reporter called me yesterday, and we talked about it, and she said, what do you really, at the end they always say, is there anything else you want to tell us? And I said, look, most people think viruses are evil and they make you sick, but I think they're beautiful. I love them. And look at this picture. It just shows how beautiful they are.
Sinister and beautiful.
So a couple of weeks ago, we talked about cryo-EM of CalP mosaic virus and how cryo-EM is great because you don't need to make crystals. And that was always the roadblock for getting structures, because you had to first get a crystal that would diffract properly, and it's really hard. And now cryo-EM is the way to go. You don't need a crystal. You just take virus particles and freeze them and take pictures, and you get 3.8 angstroms resolution.
And it's been, there's just been a revolution in the equipment, especially the sensors for it. I'm doing an article on this right now that has really, really enabled that, and that's why you suddenly see all these cryo-EM, high-resolution cryo-EM structures coming out.
Yeah.
Because once you pass the barrier of the several $1 million investment to get these instruments set up, which I think is what the Purdue press release is referring to as the additional equipment, then you can really start pounding out stuff like this and even do it on relatively short notice.
How do they pick the colors?
Exactly.
Whatever they want. Well, there's usually a convention for viral proteins that sometimes people can follow.
And I think they're following their own convention for what they did with dengue.
Yeah. Because for picornas, we give VP1 is blue, VP2 is yellow, VP3 is red, VP4 is green. And you're supposed to follow it just to be consistent. Otherwise, but you can make pastel versions and it looks prettier. I like when I made some figures for something, I don't remember. I made them pastel because it looked a lot better. Now, the thing about... So Rossman Group and Richard, they have done a lot of flavies. They've done a lot of picornaviruses. So last summer, enterovirus D68 popped up, causing a big epidemic. And within a few months, they had a cryo-EM of that virus, also in science. So I figured they were going to do this. They're going to be on it for sure. Yep. And they are. Mm-hmm. Very pretty.
And, good way to publish these days is to work on Zika.
Is that right? Do you know this virologist named Kathy Zika?
Yeah, I heard about her. I think she used to have a different name, but
it's a way to get published.
It's a cool name, actually. Kathy.
In fact, I just tweeted that paper the other day that we were discussing on email. They actually published a paper on Zika virus without doing any experiments with Zika virus. It was rather stunning.
Yes, the situation is out of control because, unlike Ebola, where most people can't work with it, right?
Right.
So you didn't have this big people doing it. Now a lot of people are working on it, which I think is fine because we need as many minds as possible. However, I wish the journals would just ask for a little more to publish, right?
Well, and I think I understand why this is evolving the way it is, but I'm a little disturbed about the long-term health of the scientific enterprise when you get this huge boom in publication and press and funding for one disease or one virus, and then 6 months from now, it's going to be something else.
Well, first of all, I don't think there is any boom in funding from what the NIH program officers have told me. Obama's $1.8 million request was rejected by the House.
Billion.
Well,
right?
yes,
billion,
there is that.
Billion,
right,
yeah.
But it was a boom in funding requests anyway.
Right, and, 'twas ever thus. Just think about HIV.
Yes.
There was a huge influx of money into HIV that has never gone back down from what I can tell. That may be an exaggeration. But
Kathy, this RFA, that Zika RFA, that's not accompanied by any extra money?
That's correct.
It's going to come out of something else, right?
Right.
No. My understanding is it's going to be the same. So the RFA, I don't know if you're talking about the rolling RFA for R21.
Yeah.
That has no additional money associated with it. So it's just the normal R21's pool.
What I meant in terms of the sustainability of it, though, is just in terms of public issue fatigue. If it keeps being the virus of the month that keeps coming up, eventually people are going to say, "Oh, they always do this," and I don't think that's healthy. I mean, yes, it is important that we study Zika virus. It's important that we study a lot of other viruses too.
I agree. I think that if this happens year after year for a number of years, they'll get tired of it. Yeah. Well, every time there's a new virus, you guys start working on it.
Yeah.
Esper, what is the mindset in Brazil at the moment?
In terms of funding?
No, just people in general. They scared, of the virus.
No, yeah, I think the main problem are those women who are planning or are pregnant. I mean, there is very little we have to say to these women, just besides buying loads and loads of bug repellents, right? Because it's very, very hard to predict. We don't have the numbers. We can't tell them what are the percentages a woman may transmit the virus to the kid. There are numbers out there ranging from 1 to 29%. So it's too much of a range to counsel someone who is pregnant. What would you do with a woman who is in the first weeks of pregnancy, have like 30 weeks in front of her, and we don't have reliable data to see where the virus is circulating more or less. So this has been a struggle. We know that many women are avoiding pregnancies. We heard cases of termination. And although you may know that besides raping or risk to the mother or anencephaly, pregnancy termination here in Brazil is considered illegal. But even so, there are hundreds of thousands of women that undergo abortion, and depending on the level of the social stratum, a woman can decide to go to a safe or a non-safe abortion if she is too much afraid, not been well informed, or if there is a detection of birth defects in some cases. We fear a lot about this type of misinformation, so we're trying to spread as much as we can what we know about the Zika virus. But we are in a very uncomfortable situation in the country.
Has this stimulated more discussion in Brazil about access to safe abortion?
Yes, there are several groups pushing this debate forward. But this is not a good time in the country for that. We're going through a political crisis that's dominating the public debate, and and it's been hard to move that discussion agenda forward.
Right.
And on top of everything, this March we had the reintroduction of H1N1 2009 strain. During our summer. This is completely unexpected. Now, if you open the newspapers, the only thing they talk about is flu. So the last few months, since the early 2015, has not been a good year virus-wise for the country.
Wow. Yeah, I heard about the influenza outbreak. That's so weird, right?
Yeah, there is no explanation why it came so early this year.
So right now you are moving into winter, is that right?
No, we are in the summer.
You're in the summer.
Yeah.
That's right. And your summer will soon end, right?
Sure. So then we go into the fall,
Right.
and we mirror your.
Yeah, we're just in spring now, right? As far as I know, right? Is that right?
It seems that way.
Well, people have been sending you shots of cherry blossoms, so I think so. Yes.
They do. send me.
Angiosperms.
Angiosperms, that's right. Yeah. I wanted to mention that in conjunction with our cryo-EM, Steen had written, your recent primer was timely. A cryo-EM structure of Zika appeared online today. I like that they provide a bit of info on the rapid review, 17 days from submission to acceptance with some sort of statistical advisor involved.
That's very fast.
Yes.
They also published excerpts of a number of letters on how easy it is to overvalue publication counts and the journal impact factor metric. Yeah, let's get rid of it.
Is it known, by the way, just as in passing, I should have raised this during the actual episode, but do we know if you've been infected once, can you get infected?
Let's ask Esper. It's a good question.
Esper,
if you get infected...
permanent immunity after you're infected?
Say that again.
Is it permanent or non-permanent immunity after you've been infected once?
No, everybody believes it's permanent.
Right.
But we still need to gather more data.
Sure.
Do you believe that it'll, in 4 or 5 years, the epidemic will stop because we will have immunized most of the population?
I think we're going to have just a few cases popping here and there, as you use all the susceptibles, and then for the virus to circulate, you need to find small pockets of susceptibles after immunity has built up.
Or howler monkeys.
Right.
Or some kind of rodents. I think there's some kind of rodents are in the mix too.
Oh.
Esper, do you think we need a vaccine?
I think we do, and the natural use of this vaccine for any person living in an area where Zika does not circulate and wants to travel to an endemic area. This is a huge clientele around the world.
Sure.
Yes.
And obviously, if you have the vaccine and the virus hits a new country, new population, then you can come and do a blockage. And,
right.
So yes, we need.
This is why you have to listen to all of TWiV, because here at the end we're getting to some key things. One other issue that we didn't bring up, Esper, I wanted your thoughts on. There is cross-reactivity between dengue and Zika virus. So do you think immune enhancement could be part of the pathogenesis?
I think needs to be demonstrated in a model first. And then we can also do that once we have a reliable serology test for Zika, which does not cross-react to dengue or other flavivirus such as yellow fever vaccination.
Yeah.
Sure.
Because if you go to areas where you had lots of microcephaly, and the serosurveys for dengue show that over 90% of people are dengue positive. So whether it's
90%, wow.
Oh yeah, like in Pernambuco, you rarely find someone who is seronegative for dengue. And then question would be, what kind of antibody would serve as an enhancement? Would be just dengue 1, 2, 3, or 4, either of them, none of them? Is the level of antibody relevant or not? And the type of antibody, there are so many questions we need to answer.
Sure.
And also, if we start using dengue vaccines, whether that would be an issue at all, right?
Sure.
Right.
Sure.
To make it worse, or maybe a Zika vaccine would make dengue worse.
But you already use yellow fever vaccine, so.
Well, yeah, but still, we have in the country about 2015, we had over 600 people dying of dengue in Brazil.
Right.
So a dengue vaccine is highly needed,
So these are people that had dengue, got over it, and then got the wrong strain the second time.
That's what we hypothesize, but in most cases we don't have that evidence present because someone coming with dengue, you're not sure if this person had dengue before based on the history. Right. The documentation is poor, and depending on the day of symptoms, you have antibodies already built up for this new infection. You cannot sort out if it was due to this infection or a previous one.
But in the area where the prevalence is 90%, and then you have a death from dengue, it's pretty certain that they had dengue before that.
Sure.
Yeah, that's what I mean.
Like if you come down to São Paulo, only 5 to 10% of people are seropositive for dengue.
Got it.
Is there cross-reactivity between yellow fever and Zika? Do we know?
I don't know.
I'm not aware of that, Dickson. That's why. So with dengue and Zika, there is. That's why I brought that up.
Right.
Right. I mean,
which could end up being a good or a bad thing.
That's right.
Some of the other flaviviruses do cross-react, like West Nile with St. Louis encephalitis and that sort of thing. But
there were two different places of cross-reactivity mentioned on the CDC site when we did an episode several weeks ago, and they had two different sets of cross-reactivity with other flaviviruses, and so I can't remember about yellow fever virus. Back to your question, Vincent, about the Zika virus, the early epidemiological and genetic findings paper with Faria as the first author, the science paper, did mention that the reported cases in Brazil peaked in mid-July of 2015, and they show a pretty impressive graph showing that. And so what we don't know is whether it'll, peak again in the coming July or not. But that at least addresses the epidemiology for almost 1 year so far in this graph.
Right.
So, Vincent, since you're bringing the key questions in the end, there's another one that I would like to raise regarding Zika, which is the Guillain-Barré pathogenesis.
Right.
Guillain-Barré is taken as a autoimmune disease caused by a post-infection creation of autoantibodies, mainly directed against myelin. But I would be very curious to see whether the virus has a direct effect on the axons in an acute phase. And I think this is an important thing that we need to sort out again as well, because in some of these cases of Guillain-Barré, the symptoms develop while the viremia is still present or the viruria is still present. And this, to me, raises the question whether the virus is damaging directly the neural cells or not.
Right. Have you seen Guillain-Barré in Brazil?
Yeah, we had one case, and this woman had symptoms starting at day 9 after onset of Zika. And on day 13, she was still Zika positive in the urine, which is very unusual for a post-infection Guillain-Barré syndrome.
Interesting. Has that been published, that case?
No, but there are two similar cases from Polynesia published.
Right.
Exactly like ours in Euro surveillance.
Got it. Someone's backing up.
Not me.
Not me.
This episode of TWiV is sponsored by the American Society for Virology, who would like you to know about this year's ASV meeting, which is being held in beautiful Blacksburg, Virginia, on the campus of Virginia Tech, June 18th through the 22nd. The local organizers are XJ Meng and Zach Edelman and their committee, and Kathy Spindler and her committee are arranging this great meeting. This year, the keynote will be given by Dr. Stephen Russell on oncolytic viral therapy, and then you'll have 5 days of symposia, workshops, poster sessions, and state-of-the-art lectures. Come listen to Dr. Joe Handelsman talk about leading the White House Office of Science and Technology Policy. Be part of a live TWiV. Yep. And enhance your presentation skills at the Career Development Workshop this year. Also, we will have at ASV a special Zika virus oral workshop and poster session. They've made a call for late-breaking Zika abstracts. Deadline is May 16th. You still have some time to get out those neuroblastomas, do some experiments. Please do some virus experiments.
Yes.
Not just
Not just RNA.
Yeah.
Richard promised that they'll have an abstract in that late-breaking Zika session.
Cool.
So.
Early registration ends on May 20th. You can get reduced rates. So go there. You go to asv2016.cpe.vt.edu or asv.org, and there's a link there as well. And I hope to see everyone there. I will be there, and Kathy and Rich Condit doing our TWiV, among other things. It is 3:26. We can do a couple of email. We have one on Zika from Stephanie. TWiV, I just started listening to TWiV today and listened to 377. The discussion was women having babies positive for Zika, but themselves having negative sera for the antibody. Is it possible that this could support the sexual transmission theory? Semen is the carrier, but possibly not able to cross between fetus and mom. You may have addressed this already. Thank you, Stephanie. Esper, what do you think?
I think the passing the placenta depends on the level of viremia of the mother. So if the semen infects the mother, it eventually can do that. I'm not aware of any case that a woman has a confirmed Zika case, microcephaly case, for example, give birth to a microcephaly case with a negative antibody.
So this case, this was the New England Journal case where the mother was antibody negative, and this is the one where they found Zika only in the brain of the fetus.
Right.
I think
you mean the first case report?
Yeah, I think that was it, right? Alan, Kathy, was that?
It was, and she was PCR negative. Was she also antibody negative?
I thought so, yeah, and that's why we remarked on it that it's pretty weird that.
Right.
I don't know how you would explain that, right? Unless the test just didn't work. Who knows?
Right.
That could be. All right. So you don't think that was correct, Esper?
I think the serology tests are under development. And,
right.
We see so many problems with cross-reactivity, but probably we may bump into low sensitivity assays. So it's very hard to tell.
Alan, can you take the next one?
Yes. So this is from Johnny, 99-cent iTech mosquito repellent. Another tool in our armamentarium. Hadn't seen this before. And she links to this mosquito device, which supposedly uses high-pitched sound waves to keep mosquitoes away. Almost silent and working frequency of 16 to 22 kilohertz. Lists some other advantages. I can tell you from looking at the research on these and also from personal experience that they don't work. I have not tried this particular one, but the concept of using ultrasound to repel mosquitoes, it doesn't work. It's a lovely idea. It would be wonderful if it did.
So this is something you put on your iPhone or iPad or something?
Yes,
Android.
I think this is some kind of either app or,
Dickson, what do you think, Dickson? High frequency? No?
Well, I know that mosquitoes are attracted to certain frequencies because I watched George Craig, who was one of my mentors at Notre Dame, who established the biology of Aedes aegypti in the laboratory. And in order to feed them, he would anesthetize his arm with them, basically by just putting it on the top of the cage.
Right.
And he said, watch all the males come over to the female. And he had a tuning fork, and he hit it on the side of his desk and held it up to the cage. And every male mosquito just gravitated towards that. So that's where the idea came from. But
it's the opposite of that.
It just does the opposite, unfortunately. Yeah.
Or it does nothing.
Or it does nothing. Or it's the wrong frequency.
Obviously.
Right. Okay. So much for that.
Yeah.
I mean, everyone would have to hold their phones up on with this thing buzzing constantly, right?
Well, or you would have it as a standalone device. If this worked, sure, I'd wear one around, but no.
Yeah. Kathy, can you take the next one, please?
From Nicole, who writes, Dear Drs. Racaniello et al., I am a longtime listener and enjoy the show immensely. I wanted to correct a statement about mother-to-child transmission of HIV-1 made towards the end of TWiV 375. I'm a few episodes behind. It was stated that HIV-1 does not cross the placenta and that mother-to-child transmission of HIV-1 occurs during labor and delivery. Mother-to-child transmission of HIV-1 occurs during 3 distinct time periods in utero, 5 to 10%, intrapartum, 10 to 20%, and postpartum through breast milk, 5 to 15% of untreated pregnancies and breastfeeding periods. And the infants have strikingly different outcomes, with 50% of infants infected in utero dying of their infections by 208 days of life versus 380 days of life in infants infected intrapartum. Over 50% of infants infected via breast milk are still alive after their first birthday. It is hypothesized that the different outcomes are likely due to differences in the immunologic state of the fetus, newborn, and infant, though differences in viral factors may contribute as well. It should be noted that while in utero transmission of HIV-1 does occur, it is not known how much is due to bidirectional cellular trafficking between mother and fetus versus exposure to virions. Risk increases with maternal viral load, maternal infection acquired during pregnancy, inflammation, prematurity, etc. Also of note, while infants are exposed to billions and billions of virions potentially in utero and through the breastfeeding period, 80% of infants escape infection. The mechanisms by which most infants escape infection is an area of ongoing study and likely is related to a lack of activated target cells in the infant and the infant immune system being more tolerogenic by design. Kathy Spindler touched on some of this interesting research by Mike McCune at UCSF in one of the discussions. Carolyn Coyne was a wonderful guest and highlighted the paucity of data about the placenta. There are many excellent researchers investigating mother-to-child transmission and infant immune development, highlighting that the immune development of the infant has to be different by design. More discussion is in the manuscript below. It is currently 71 degrees Fahrenheit, 22 degrees Celsius in Los Angeles, with humidity at 41% and wind of 2 kilometers per hour. Keep up the good work. Sincerely, Nicole. And I should have noted that all through the letter she had references, and then she's got the numbered references down below.
Just like a good manuscript, right?
Yes.
Yep. Yeah.
All right, so that was an episode with Carolyn, Helen, and Jeremy Luban. So, I didn't say that it didn't cross the placenta, but I guess in utero infection. So all of them agreed with it. So,
right,
right. Esper, does HIV cross the placenta or infect in utero 5 to 10% of the time?
Yeah, and in fact, this percentage has changed over time.
Yeah.
In the beginning of early AIDS days, most of the cases were perinatal in breast milk feeding.
Got it.
And as the antiretrovirals came on board, it shifted everything towards in utero transmission. And the reason is because basically the adequate use of antiretrovirals during pregnancy and after labor have dramatically cut down the number of vertical transmissions. So the in utero transmission was left in the first wave after antiretroviral therapy for perinatal transmission came on board. And then later on, we usually, in most of the countries that have resources, we've been providing antiretroviral treatment for every single person who are viremic. In that case, you also prevent transmission during pregnancy by making a woman who is pregnant undetectable for HIV virus load. So these are dynamic numbers. They change over time depending on the conditions.
I see.
The transmission in utero happens mostly, as she pointed out in her fine email, in cases where women have other pregnancy problems. And usually that leads to an inflammation of the placenta. It becomes more permeable to the virus and then eventually gets to the fetus.
Got it. Okay. So normally, in a normal placenta, you would not have crossing of either virus or virus-infected cells.
That's correct. However, there are she mentioned Mike McCune, but our group, and several other groups have demonstrated that a significant proportion of kids born to HIV-infected mothers, they have some type of T-cell-based antigen-specific immune response against HIV. And this is probably in line with her point, saying that sometimes the kid may have contact with virus products,
Right.
virus epitope, or even a complete virion. We don't actually know, and this is an evolving subject. Very interesting to explore.
Dickson, can you read an email?
Tom writes, Dear Vincent and the Merry TWiXters. That's a nice name for us. In Austin this morning, it's 20.6 degrees centigrade, 69 degrees Fahrenheit, overcast and drizzling with almost no wind. The predicted high is 28C, 83F, with a chance of a morning thunderstorm. So, I was disheartened to hear Vincent berating himself for the use of the word so. I fear it was caused by my letter some time ago commenting on how it was often the first word in someone's response to a question. It was not meant as a criticism, just as a curiosity. I was referring to its appearance as a standalone expression at the beginning of a paragraph. So I was, that was my mistake. I was not talking about its use to indicate the reason for an action, as in we used fluorescent tagging so we could observe the gene's expression. Since then, I found myself reading Seamus Henley's wonderful 2000 translation of Beowulf. In the introduction, he commented on the challenge of translating the very first word of the Old English epic. He writes, conventional renderings of thwaite, the first word of the poem, tends towards the archaic literary, lo or hark, behold, and attend. The particle so came naturally to the rescue because it functions as an exclamation, calling for immediate attention. So relax.
I think it's flat.
And use so as you've been. You're doing it right.
Yes.
The renderings of the...
Oh, is that how that is pronounced? Right. He writes further, I appreciate the Zika reports. We've moved into the country outside of Austin where my wife's CFS symptoms have very much improved, but now I'm acutely aware of mosquitoes. With our unusually mild winter, April came in February, and what looks like a wet spring and summer, we're already seeing lots of mosquitoes. I have been repairing all of the screens, but we needed some kind of BSL enclosure to prevent them from coming in every time we go out or in. On the upside, the mosquitoes so far have been very small species. Very, very small species? And hopefully not a Zika transmitter. They're very small. P.S. I should have foreseen Alan Dove's sax symbol pun, but I suspect that he could have punned on any instrument I assigned to him. I wonder if band directors were told to be nice to Django.
Who's that?
Reference to Django Reinhardt, by the way. A French guitar player that played with Stepan Grappelli during the occupation of Paris. And then later on became very famous in jazz circles at Café La Haute.
Wow, I'm impressed, Dickson.
Any more information you want?
No. Someone on Twitter asked me when I introduced Alan to play a little yakety yak.
Yakety sax.
Yakety sax? Is that different?
Yes, it is different. Yakety yak is, I guess, a rock song. Right. No, yakety sax.
Boots Randolph, wasn't it?
The standard comedic. So this came out of a Twitter conversation. Somebody posted, said, baseball players have walk-up music that plays when they're walking up to the plate. If as a scientist, what would your walk-up music be as you're about to give a talk? And I said, well, if it's based on my lab work, it would have to be yakety sax.
So I could find that and put it in front of you.
You can Google yakety sax and you will immediately get a recording of it, and you will immediately recognize it because it is the it was the theme song to the Benny Hill show.
Oh, okay.
And it's yes, exactly. Yes.
All right.
The comedic.
I'll do it. I'll find it and put it in. Okay.
Yes.
At the top of the show. There's one more short one from Tom. Same Tom in Austin.
Yeah, same Tom, right?
That's the one you were starting.
It was.
Today's Austin Chronicle listed the Austin Music Award winners, part of South by Southwest. The Best Cover Band Award went to a group named More Cowbell.
Right.
Which has come up on TWiV before, Dickson.
I understand that.
Do you remember?
I do.
Said,
don't pin me down.
More cowbell.
Right.
Okay. Let's do some picks of the week.
Good idea.
What is your pick, Dickson?
Well, you didn't put it in there, but I sent it to you.
All right, I'll put it
That
in, right?
happens every time with you. I don't know.
I would really
Don't
like
get it.
you to put your own picks in like everyone else does.
I did. I mean, you can just stick it right in there. I see what you mean.
That's called a editable Google
Okay.
doc.
I haven't done that yet, but I'll learn how. It's
Really?
a review of
I'm going to
mosquito
paste
control.
it in. I'm going to paste it in so everyone else can look at
It's
it.
a very good overview of the potential control mechanisms in place now for limiting populations of mosquitoes, including some genetic manipulations as well. Nice. It appeared on NPR, in fact.
A starry-eyed four-step guide to wiping out a mosquito.
Yeah.
And
Nice.
Dixon,
It's a good review.
we need a world mosquito czar. I think that should be you.
No, I think it should be Tony.
Anderson, what's his name?
No, I'll
At Irvine, right?
yeah.
Tony James.
Tony James.
Yep.
He would be perfect.
Step 2, figure out the best way to wipe them out.
Yeah.
I think you get stuck on that for a while.
Yeah, although I'm sure Dwayne Gubler would step forward to be that
Step
person.
3, raise the money.
Right, that's a big one.
And step 4, eliminate.
Yeah, and then he's got a lot of...
No, first you get the money. Then you get the... Oh, no. Sorry, wrong script.
Then they can't find you for 3 months.
Right.
All right.
Yeah, this has been tried before.
No, I'm not saying it's new. It's just a nice summary of... Because we've
It is.
come into vector control
Yes,
discussions before.
and I like the tag of it as a starry-eyed for it. Like this in an ideal world. Here's what
Yeah, well, haha. Good luck on that.
Alan, what do you have?
I have a video game, it's been released. I found out about it a few weeks ago. They had released the artwork for it, and then I just found out that they finally released the initial version online. It is currently only in Portuguese.
That's for Esper.
Esper.
For you, Esper.
But it's Science Combat. It's based on the game Mortal Kombat, which I don't know how many people ever played that, but it was an 80s video game. Anyway, it's all done in 16-bit 1980s style, in keeping with the aesthetic of the original game. But you're playing as a scientist, as one of several scientists you can choose. Of course, I chose Darwin, and you get matched up one-on-one, two-dimensional fights with Nikola Tesla and Albert Einstein and various other folks. And each scientist has their own special moves that they can do.
Yeah.
So once you figure out the controls, as I say, it's in Portuguese, but it's very easy to figure out. It's your basic button mashing action game. And after you finished off all the scientists, there's a final boss fight that's pretty entertaining too.
Alan, I understand the English translation to that is Portuguese man of war. Just a little joke.
Yes.
Very, very little.
No, come on.
Kathy, what do you have?
And it says, Alan, it says it's dedicated to old video games and the best scientists of our history. That's
Great.
what it says in there. Yeah.
Okay.
Good.
Kathy, what do you have?
So I picked something that was in the Purdue press release about the 3.8 angstrom resolution cryo-EM structure of Zika virus that I didn't see in any of the other media outlets that came up with this story. And this is a YouTube video that has no audio to it, so if you haven't looked at it, you can go look at it now. It's just beautiful. It shows the cryo-EM structure in a number of different views. It really focuses in on the glycan binding near the asparagine 154 glycosylation site, and it's really lovely. And this is why I mentioned the third author, Lei Sun, because he's credited with putting this video together. And so, go Purdue.
Yes.
I wonder why they have the video unlisted on YouTube.
I don't know. I saw that too. It's a Purdue thing, and evidently maybe just something they didn't check or click
Yeah,
or
sometimes that
something when
happened,
they posted it.
Because it's good to share.
Yeah.
I'm going to add it to one of my collections called Viruses. Okay, that's very cool. There's also a video on the Wall Street Journal of Rich Kuhn and Michael Rossman and the young woman doing the cell culture, the first author there, which is also
Dickson.
interesting.
Yeah.
Check that out as well. Esper, what do you have?
Yeah, I selected a poem that I received from a friend. His name is William Bonnet. He works at the University of Rochester. And the poem is called The Chaos. It's a poem written by Gerard Nolst Trinité, who is a Dutch writer, teacher, and traveler who died in 1946. And he was called by the name Carivarius. So this appeared in his book called Drop Your Foreign Accent. And I pasted the YouTube video there so you can follow someone dictating the poem. And this is so you have an idea how hard it is for us who are not native speakers to understand sometimes what you're saying. The pronunciation clues and tips. And so it's a very nice poem, and I found it fascinating and
it's really good. I watched the whole video. It's got a running script of the words, so you can see that he's mostly focusing on words that are spelled the same but pronounced differently. And there are, just a large number of them. It's really good.
Yeah, I don't think we've had a poem ever picked on TWiV, have we? Maybe a long time ago. It's been a lot of picks, right? I might forget easily since my memory is fading. Cool, thank you. And you got something else too, right?
Yeah, well, this is a self-serving pick. We run every year a HIV pathogenesis course here in Sao Paulo. The next one is coming up. It's going to be from April 14 to 20. And we have a very good gathering here with lots of colleagues from the United States mostly that come to the University of Sao Paulo to teach the students. So the course is free. Anyone who want to attend the course can register through the website where they can find the program. And the speakers they come on their own to Brazil to collaborate with this initiative that we are going to the 11th edition. So I thought I could have a TWiV bump and get this course famous around the world after this episode.
You bet. Everybody will be coming there to take it.
You better pick a bigger auditorium.
Yeah. Carl Zimmer hit 250,000 Twitter followers after he was on.
Is that
and before he had 249,000. No, he had less. He had less than that. I don't know.
That was a TWiV mosquito bump.
Mosquito bump, exactly. Thank you, Esper. My pick is an article in Outbreak News today about the results of a Harvard poll, which was done on a number of U.S. households where someone is pregnant or considering getting pregnant, and asking them about Zika. It was done by the Harvard School of Public Health, 1,275 adults. I didn't actually look at the original survey. I don't know. I didn't Google it, and it's not linked in this article. So this is a summary of it. The headline reveals public's lack of information about Zika, pregnancy, and sexual transmission. And of course, the sexual transmission got me here. Approximately 1 in 4 are not aware of the association between Zika and birth defect. I'm glad they used association. That's good. 1 in 5 believe that there is a vaccine. 4 in 10 do not realize Zika virus can be sexually transmitted.
How would you have answered that question?
Well, I would like to know how they posed it.
Yes.
Because as Kathy said this week, doing surveys is an art in itself.
Yes.
And, they probably said, is Zika virus sexually transmitted? Yes or no, right? I would guess that's the simplest way to do that, but who knows? But I would have...
And I might have answered no. I mean,
that I realize it.
Yeah, depending on how you ask it. Yeah, exactly.
That's true.
That's a key thing. And I think they shouldn't say, it's sexually transmitted, because as we've said, it's not clear. And if it is, probably really the byway, not the
it's
highway.
It's not primarily, right.
And so, yeah, I'm not surprised that people don't have the latest Zika information. And TWiV is too long for most people to listen to. I understand that. Now, that, of course, is the job of CDC and local agencies,
CDC,
right?
Usually,
a lot of people don't even trust CDC. You know, I tell often people, go look at the CDC website. We don't trust them there.
Right.
We have 2 listener picks. One is from Justin, who sends a link to a blog post about an article that appeared in the Journal of Virology. He says, really awesome paper, especially as a mass spec chemist. It's great to see how it's become such a powerful tool for even virus discovery. This is a paper called Acidianus tailed spindle virus, a new archaeal large-tailed spindle virus discovered by culture-independent methods. It's a cool story of how they found in Yellowstone Park they were looking at environmental metagenomes, and they found a virus sequence, and they designed primers to try and purify it from actual specimens, and they were able to track its purification and get it. I was thinking maybe we have the senior author, Mark Young, come on and talk about that. It's a cool story. Yes, it's a very cool story. And then we have one from Melinda. Here are 2 picks. First, Dave O'Connor at the University of Wisconsin-Madison has infected some rhesus macaques with different Zika isolates and is reporting the data in real time at the Zika Open Research Portal. And she provides a link to that. And you have some comment on that, Esper, right?
Yeah, so Dave is a collaborator since 2005, and we've been working on the GBVC, SIV/HIV co-infection aspect. Well, I got him interested in also studying GBVC because he went to Uganda, to the Kibale Park, in a project to find new virus. It's a virus discovery project. And in his search, he found lots of monkeys infected with GBVC. And based on this finding, he decided to move ahead and establish the animal model for GBVC infection. That's Adam Bailey's paper that you mentioned a few times here in previous TWiV episodes. And one of the things that caught their attention was that during the Ebola epidemic, the Broad Institute decided to put all their data online. And having access to the data the Broad Institute had on virus discovery out of actually was pathogen discovery out of the Ebola cases from one of the 3 countries affected in East Africa, they also described the association between GBVC infection and lower rate of fatal outcomes of Ebola. This is just a cross-sectional finding. It was mentioned here in TWiV as well. But that experience was really instigating because having data put online in a medical emergency, such as Ebola was, or such as Zika has been, it's something we need to discuss more. And that was his inspiration to decide to ask for a supplement in some of his grants and start doing this Zika model up in Madison with a group of very young and bright researchers. And what he decided to do was to put all the data he was collecting online real time. We must all be aware that this is not peer reviewed, but it's very important because in situations like this, having access to data as fast as we can would save potentially lots of lives down the line if you speed up the process of communicating your results.
Right.
The results have been compiled. I think they're working in the first manuscript already to be submitted, but the experiments that are coming up already are making their way to the website, and people can follow them like a real like real-time story, and this is very instructive to whoever is working on Zika. Not only people who are trying to put up monkey models, other animal models, but even us who are clinicians who can take some lessons from these experiments, trying to see if they match what we see in the clinic. So it's very valuable experience.
Cool.
Also, what we brought up in this one-page comment is that in times like this, we need to be more open in sharing samples, sharing information, and sharing specimens. And we know this has been a little bit of a problem in this epidemic. We're learning from it, and I think it's a great opportunity to improve in that direction as well.
Yeah, it's something that's always a problem in science, and still, even as you say, people are not sharing Zika and reagents, and sometimes it's because the universities want you to do MTAs, and that takes a long time. And I'm trying to get a virus from someone, and it's been weeks because the lawyers are arguing back and forth. It's just crazy stuff. Melinda has another pick. Great book for kids, Your Body Battles a ... series by Vicky Cobb. And she's got quite a few books. Your Body Battles a Stomachache, Your Body Battles a Broken Bone, a Cavity, a Vertical Farm.
No, there's Your Body Battles a Cold, though.
An earache.
There's no Vertical Farm?
No.
I wanted to see if Dickson was awake.
Well, he's awake, all right.
Anyway,
I'm just waiting for the right moment.
Talk about taking advantage. I mean, some of these used copies, $400. Come on. It's a little bit much for a used book.
Pretty rare.
I mean, it's just out of print, I guess, and people take advantage of these things. It's all about money, Dickson.
Those are the book sellers. That's not the author.
Right, that's the price being asked. That's not necessarily
they don't get
the
that
price
price.
they're going to get, because there's one.
No,
there
it's
are
not
others.
the author. I understand, but it's someone who happens to have a copy.
It's a bookseller.
Right,
No,
but
it's
then
not. It's just a person.
There are additional copies right underneath that for like $30.
Yeah, I understand.
So
But
Or
they're
12.
not going to sell them.
it's bothersome.
One of my copies for Parasitic Diseases 4th edition was listed under one of these things for $1,600. I said if I had 3 more of those, I could take a vacation. But, of course, no one bought it for that much, and I wouldn't have got that money anyway.
No, it's a third party. All right, thank you. Melinda is a professor in the University of Georgia, and probably too young for Kathy to know her.
Well, she may have come after I did,
Yeah,
but
she's an assistant professor.
I may meet her next week when I'm at the Southeast Regional Virology meeting.
In Georgia?
Mm-hmm.
All right.
Going down for the the golf tournament?
It's the same week as the Masters, indeed.
I'll be darned.
Yep.
This episode of TWiV 383 can be found at iTunes and also at microbe.tv/TWiV. And we love getting your questions and comments. Please send them to twiv@microbe.tv. Dickson Despommier is at verticalfarm.com, the father of the vertical farm. Thank you, Dickson.
You're welcome, Vincent. And by the way, anybody who wants to see more about the Vertical Farm can see last Sunday's New York Times business section. Has a nice article about vertical farming.
Why don't you send people to urban agriculture like that? The podcast about vertical farms, Dickson.
We'll talk after.
In fact, this was an interview of a vertical farm that we did last June on urban agriculture.
That is
right, Dickson?
Yeah.
2 entrepreneurial women. It was a really good story, and I had it as my pick until the Purdue video of the Zika virus came out.
We'll make it for your next week's pick.
We interviewed.
I was going to, but you already mentioned it, so we can't.
We interviewed them last June, Kathy.
We did.
Yeah,
cool.
It was a lot of fun because the architect designed my colleague's home out there in Wyoming. So we know her. All right. Thank you, Dickson. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
Alan Dove is at turbidplaque.com, and you can also find him on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
Our guest today is Esper Kallas. You can find him at the University of São Paulo or on Twitter. He's eKallas. EKALLAS. Thank you, Esper.
It was a delight. Thank you. It was a great experience for me.
Great having you, and you should come back sometime.
Thank you.
All
Absolutely.
Right. We'd love to have you back. And, this Zika story is going to continue for a while. Brazil is the epicenter, so you should come back sometime and help sort it out again.
Definitely.
Do that.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.